Okay, so last week we looked at the Calophoridae, the most important family in Diptera that shows up on dead people. Now we're going to move into a couple of other families that can cause some issues. The first and foremost are the Sarcophagidae. These are the flesh flies. So flesh flies, these are those black and white checkerboard and striped flies that you see flying around. And I talk about these second just after Calophoridaes because they show up a lot on bodies. So the adults are medium to large size flies. They range from about 2 to 14 millimeters in length. So they're really large. They have these gray and black vitti on their thorax. And then they have what we call tessellated or checkerboard pattern on their abdomen. So this is how we can tell the difference between the different major adults. Is in Calophoridae, remember, they tend to be metallic. In Sarcophagidae, they got this tessellated thing going on there. So they're usually black and gray. All right. Now, the other major characteristics, though, that we can tell Sarcophagidae from Calophoridae is the arista of the antennae is plumose only at the base. So if you look here, here's the arista, right? Remember in the Calophoridae, it was all the way to the tip, but this is only plumose here. And then notice it goes smooth at the tip. So it's only plumose at the base, where it's plumose throughout in the Calophorids. And the bodies of the Sarcophagidae tend to be very bristly. The eyes are widely separated in both sexes. And in some species, the eyes are bright, bright red. They also have these highly visible genitalia at the tip of their abdomen. And that gives them the common name of the red-tailed flesh fly. Now, the larvae of flesh flies are really easy to tell the species apart too, because they have these posterior sphericals that are in this pit or this depression right there. So instead of being flush with the surface of the posterior segment, they're in this little pit. Sometimes this pit closes, so you can't even see the sphericals. So this is really easy to tell even in the field, and the maggots are huge. Just pick up the maggots, and if it's a pit, you know it's Sarcophagidae. Very, very simple. Now, this character is probably the easiest just to use on sight, this deep pit. But if you get them underneath a microscope, you can look at the spherular openings. Remember when we looked at the Calophoridae, they had their sphericals where the spherular openings were pointing towards the midline. In Sarcophagidae, the spherular openings are pointing up and down or away from the midline in the case of this first one. So they have a different orientation and they're in that deep pit. Super easy to tell the difference. Now, if species idea is necessary, this is difficult with sarcs. So the species of Sarcophagidae are very, very similar to one another in both the adult and the larval stages. They're extremely hard to identify at the moment. We don't have any really good keys out there. In fact, there's only a handful of people that know Sarcophagidae well enough to really be able to identify them morphologically. So if the species idea is necessary, you should rear out the maggots to adulthood and then find some of these people who actually know what they're talking about. Now, the basic life cycle of the sarcs. Once again, sarcs are holometabolous. They have that complete metamorphosis. So these flesh flies are attracted to carrion under most conditions. They will oviposit in the sun, in shade, in dry or wet condition, indoors and outdoors. So because they're so large, they can take a lot of different environments. They can do a lot of things. Now, they're frequently found on any decomposing tissues located within an indoor environment. They're really good at getting inside. So this makes it kind of unfortunate if you're dealing with a lot of indoor, say, homicides or deaths, and you show up with sarcs. It can be very hard to do time of colonization estimation with these species. They can also be found associated with carcasses throughout early and late stages of decomposition. So those blending waves of maggots that show up, blending waves of different species isn't as pronounced with these sarcs, so we see them all over the place. There are some species that prefer really tiny carrion. So there are certain Sarcophagidae that will only oviposit on things like dead insects or snails, while there's others that feed exclusively on larger vertebrates. So we do see a differentiation there. There's also a few species that act as parasites of things like Hymenoptera. So there is one species that is a parasite, an obligate parasite of Hymenoptera. So they're all over the map with these, with their hosts or what they feed on. But regardless, Sarcophagidae tend to be larviparous. Now, what they do is they will retain eggs in their abdomen until the eggs hatch. Then they're going to deposit the living first instar larvae on the decomposing remains. Now, when we use Sarcophagidae as a time of colonization estimator, we need to eliminate the egg hatch time then, because they're only depositing the larvae. Under a few conditions, however, they can deposit eggs. We're not quite sure what causes that switch. There's been a few papers out recently on oviposition tendencies, the Sarcophagidae, so it is possible. But we don't know, is it a genetic thing? Is it an environmental thing? Is it just a few of them happen to do this? But in most of our developmental studies, the ones that we actually have, there is no egg stage because these are considered overall larviparous. Now, the larvae may be cannibalistic, so they may eat each other in really large instances or really large maggot masses, but they do tend to focus on younger, smaller larvae that happen to wander in front of their little mouth hooks. They don't go chasing after anything else necessarily. Now, just like other flies, the larvae are going to feed on this resource for however long it takes. They're going to then leave the resource and find a place to pupate, and then they're going to eclose into the adult fly. Now, adult identification. This is primarily accomplished in the male Sarcophagidae. So the male and female Sarcophagidae have what we call a lock and key mechanism of mating. This means that the males have an incredibly complex genitalia. We call this genitalia the adiagus. So the adiagus will have all manner of different bits and bobs hanging off, and it all looks significantly weird and different, and there's all these things. So this adiagus then is the quote-unquote key, and it fits into the female's incredibly complex vaginal opening. This complex structure ensures that only members of the same species will successfully mate. So if you look at every one of these adiagi right here, these are all specific. There's a counterpart in the female where it's just basically the reverse of this in an opening where these things fit together perfectly. But since they are so different, you can see that there is no way one species is going to accidentally mate with another. It just physically will not fit. So this is how we have to tell our species apart at the moment. This adiagus shape, best way to tell the species of Sarcophagidae. So if you end up with a female, it may be impossible to identify using current keys. So looking at keys, they look exactly like this slide. You'll get all these pictures with all these crazy drawings and all these names of these different parts on here, and you basically match up the adiagus on your specimen with what you see in the key. Females, you just can't do that right now, so it may be impossible to key them out. In fact, this is something that they test you on the ABFE exam, the American Board of Forensic Entomology exam. Part of the exam is identification. And when I took the exam, they put a female Sarcophagid on there and said, what is it? And I had to say it's Sarcophagidae. That's all I know because it's female. And so that is what they're looking for. Now you know. Now, there's only a few Sarcophagidae experts in the world, like I said, at the moment. So it's probably because it's such a pain in the butt to identify Sarcophagidae. When you identify these things, what you have to do is you take the specimen and then you have to spread out the adiagus. And that's incredibly delicate work. You don't want to break the thing off. And so you have to soften up the insect, you spread everything out, you make sure you can see all the parts, you pin it, and then you look at it under the microscope. So it's very, very delicate work and it can easily be messed up. Now, sarcs therefore really aren't used as extensively as caliphorids in forensics, even though they show up very quickly after death. And part of that problem, maybe it's just a pain in the butt to identify them correctly. This might get easier if we're able to do field DNA, but at the moment we just can't. Now, there is one species that we tend to see most commonly on dead bodies, and flies in the genus Sarcophaga, or Sarcophaga, depending on who you're going to talk to, arrive concurrently or slightly after the blowflies on carrion. Now, they're known to fly under inclement conditions that would prevent the flight of most other flies. So when it's raining, when it's really cold, they might be the first species to arrive on carrion. Now, adults commonly enter indoor habitats. Larvae are often recovered from remains located in homes. This is one of the reasons that they're really good at getting into homes is because they're able to larvaposit. So what these females will do is they'll land on window screens. If the window is open, they can smell that decomposing body inside, right? And so they'll just put their little larvapositor, their ovipositor, through the openings in the screen and just shoot larvae onto the body. So they're able to colonize these things that other oviparous species are unable to get to. Now, this is the major genus that we're going to see, especially in the US. We mostly see it in summer months, but it is very common all over the place. Now, the main species is Sarcophaga haemorrhoidalis. This is commonly known as the red-tailed flesh fly. It is known like that because the Aediagus is very, very prominent in the males, and it's just taking up what looks like several segments on that last abdominal segment, very complex, and it's usually bright red. You can just see it in the field. So it's very simple to see. Now, this species is cosmopolitan, so it occurs worldwide, and it's very large. It ranges from 8 to 14 millimeters in size, and it appears very similar to flies in the family Muscidae. It is typically larger than musket flies, though, than the house flies. And it has that obvious external genitalia, which makes it very easy to just site ID. The body is black, but it's covered in a whitish powder that gives it that ash gray appearance like we saw in some of those Caliphoridae. And it does have 3 vidi on the thorax while the abdomen is tessellated. So this, when you see the muskets over the next couple of lessons, they also have vidi, but they have 4 vidi. So we got 3 vidi in the Sarcophaga and 4 vidi in the Muscidae. The adults are attracted to both excrement and carrion, and females will enter dwellings, deposit larvae, and be able to get to these indoor bodies very easily. They're most active during the summer months, so they really like that warm time of year. Now, this species is larviparous. It deposits living first instar larvae rather than eggs, at least for the most part. And this species is known to infest wounds and the digestive tracts of living animals, especially mammals. In fact, it's been recorded completing development entirely by living within a digestive tract, so it causes a certain type of myiasis, which I'll talk about a little later. All right, that's it for the Sarcophagidae. Let me know if you have any questions.